Martin. We have a few things to discuss from the regulator's perspective here in just a few minutes. But let's move on to what I consider to be maybe, if there was a scale of difficulty, this is like 10 out of 10. Go to Susan Ward. We have enough difficulty with heterogeneity in large trials, but Dr. Ward is going to discuss overcoming variance due to heterogeneity in rare disease. Quite a challenge.
Approaches too, I think. I haven't heard any disclosures today, but I'd like to make one. It's usually financial. I don't have any of those, but I am not a statistician. So let's set expectations appropriately. The person who is James Signarovich. He's a vice president at the analysis group, and he's been my partner for the last year or so in putting together this TAP collaboration that I'm going to tell you about. First of all, why do we need yet another consortium, you might ask? Secondly, I'd like to show you just examples of results. This is not intended to be a scientific presentation. And then I'd like to talk about differences to consortia. So, and I'm talking here about Duchenne muscular dystrophy as an example. I think it's really an example. It's not a sole, independent event. So the problem is fairly clear from those little red no's, is that we just don't have clinical trials that are meeting their endpoints. Oops, what am I doing? And one of the things you'll see there is that the standard deviation, this is 6-minute walk distance, my apologies, is the primary endpoint. On average, you see a drop of 40 to 60 meters a year, but the deviation is very high. So it was only after most of the trials had been either conducted or designed and were underway that these types of pictures emerged. So these are individual patient data. The one on the left is from phase 2 Muscular trial, which also did not meet its primary endpoint. The one on the bottom right is a more recent one from the imaging DMD registry. What you can see is what one of our team members has called a hairball. And it's clearly, we've got high heterogeneity across age. So there's no wonder when you think about it that variance was very high. Mean change from baseline with a picture like this is always going to be a mess. So how can you overcome it? Well, these are the things that are typically thought of, and the bottom one there has indeed been suggested by regulators, and it did not go down well with the patient community. There was some attempt, however, actually in all of those. But the one thing I want to talk about briefly is there was a proposal around prognostic factors. And you can see by those yellow lines that making subgroups before the age of 7 or after, before kids seem to be able to walk further, after 7 they're more inclined to be stable or to lose the ability to walk. If at the beginning of your trial a boy is below 350 meters, he's more likely to decline than not. So if we look at the reduction in variance that you get from MMRM with that type of maneuver, you get some benefit, 90 meters down to 82, but not a great deal. So this is when I entered, James and I entered the picture. 5 years of clinical development, many patients, more than 1,000 open port biopsies, of which, 30 to 40% were placebo patients who will never get that muscle back. No drug approvals in 2014, and the best we'd done was 10, to be generous, 15% in reducing variance. So the question we put to ourselves is, can we learn from other diseases and other disciplines and bring something new to the picture? So the premise we developed here is that imagine each patient, as they do, as their own distinctive longitudinal trajectory. When you do a clinical trial, you're getting a little window into that. But if you have a natural history over a long period of time, as in these registries, you've got a composite of trajectories. That puts us in a position with a testable hypothesis, though this isn't quite a hypothesis. And that is, if we could focus on the longitudinal trajectory itself, describe it, we might be in a position where we could reduce variance and be able to look at distribution across the population, compare cohorts, etc. So we put together this collaboration, and this is what it looked like in the beginning. It's evolved, as things do. First of all, we were looking to see if we could reduce variance by using latent class trajectory analysis. I'll describe that in a minute. If we did, we wanted to validate it and then translate it into benefit for drug trials. That is all viewed as part of a pre-competitive collaboration. After that, the idea is we'd enable sponsors who would then go off and do their own proprietary things. And we wanted to make sure that this is communicated broadly. We will publish. We want to engage regulators along the way. That's kind of how it is. So this is the methodology that we started out with. And you're probably familiar with it, but it was developed mostly in social sciences and healthcare economics, where they have to handle highly heterogeneous longitudinal trajectories as a matter of course. And it does quite a good job of that. I did a recent search in PubMed, and it's sort of got an exponential rise, which I think is quite interesting. I got some references available. Okay, so we decided to do a rough proof of concept using publicly available data. By tracing from the publications. So this is a picture of several individual patients from several different databases. As you can see, it's color-coded. As you can see, everything's on top of each other. This is the method. The way this works is you model, assuming that everybody is around a mean, a single mean. And then you do it again and imagine it's around 2, around 3 or 4 or 5 different classes. And what you're looking for is where you optimally reduce variance. So on the left here, you can see that variance in this particular example drops from 70 meters to just below 45, actually 43. And you'll see that the lowest there is 3 classes. 3 is better fit than 1 or 2. And the item on the right is a little test to cross-check, and I won't go into that. So that's how it works. Let me show you what you can learn from it. So here is an example of that atalurin placebo arm that you saw before. And what you see is 2 clusters and a pretty dramatic drop in variance. If you recall, there's that hypothesis of prognosis around above or below 350 meters on the 6-minute walk distance. So question mark, what does that look like? And even though it was a relatively small sample, we still saw 2 clusters emerging from that. And then to bump up the number, we took additional data from imaging DMD and now sort of replicated and found that we'd reintroduced that lower one, 3 clusters, and that was 71 meters down to 43. So this is pretty powerful. Now that's only a preliminary proof of concept, but it was enough for us to say, let's really go about and test this properly. And that's a talk for a different day. But the point is that this was enough for us to want to continue. So more or less a year ago is when we really made the commitment to put it together, although legally it didn't happen until later. We now have, in this first year, we put together we have data from 2 big clinical registries and 2 large clinical practices. The number of patients, US and EU, is about 50-50. We now have more than 1,260 patients, which is, for Duchenne, is a huge database. I don't think anybody's been able to build that before. Most of which we have the dystrophin genotype characterized. Adds up to 5,000 or more than patient years. So it's a lot of data. And I quit counting at around 35,000 data points. The reason there are so many data points is because of this. There's so much more data that we were able to bring in than just 6-minute walk distance. And that raised a question. Remember that the prognostic model was based on age, ±7, and this divide at 350 meters. So, it is distracting. So we asked the question though, maybe with all these additional parameters, might we be able to take a traditional approach, just what you'd normally do, and improve prognosis? And here is a different data set from any of the others you've seen so far. We were able to build a model. This is preliminary, of course. Standard deviation goes down a lot, comparable to what we got with clustering. And, well, you can see the picture of predicted versus observed. So I guess there's a lesson there, which is let's not assume that everybody already exhausted what's there. What does it really mean? Well, this is, I'm sure, familiar to all of you. This is just a power table. It's built on one of the trials that was out there. Over on the right, you'll see that typically people are looking for a 30-minute treatment effect down here on the left. If you've got unexplained variation of around 90 meters, which you do, you've only got a 69% power. And if you're off even a little bit, you get into the red zone. Now, if we're able to consistently move that variation down to about down to 60 meters of unexplained across the top, now you've got a really confident power, and even if you're off at the worst, you would still be 20 meters able to collect, whereas with 90 meters, you'd have still only gotten 30. So I'm going to race to the end here. I think lots of applications, if you can do this consistently. Because this is the workshop, I wanted to raise that there's a tension here between the value of this kind of work to the drug companies, and that's why they joined the collaboration. They're all very enthusiastic, readily signed up, versus regulatory risk. They don't want to be guinea pigs, as you said this morning. So I think there are several places that need to be looked at. Non-traditional approaches, this is just one of them. We could look more broadly. I think there are many. It would be good to talk about how that gets dealt with. There's all the way from safe zones to stuff that's regulated, and that needs to be, I think, a joint path forwards. And finally, as you said in introduction, Duchenne happens to be heterogeneous rate of disease progression, but it's not just rare diseases, it's prevalent diseases as well. And it's really the basis of precision medicine. So if we can solve problems in one space, question mark, can we leverage them elsewhere? I'm going to stop there and just leave you with that to look at. Thank you.